Welcome back to the deep dive. Today we are doing something a little different. We're shifting gears completely.
We really are.
If you've been with us for the last 27 dives, you know we have been absolutely obsessed with action. We've been acting like mechanics, staring at an engine that is revving at 6,000 RPM. We've looked at how cells burn fuel, how they weld broken DNA back together.
How they manufacture proteins, send signals. It has all been about the doing.
Exactly. It's all been about movement, speed, catalysis. Very, very loud, metabolically speaking.
It really has.
Yeah.
We've been studying the machinery of life in full motion. The kinetics, the catalysis, the sheer speed of it all.
But today we are stopping the machine. Or maybe, more accurately, we're looking at the brakes. We're looking at the immense, and I mean immense, power of silence.
I like that framing. Because in biology, silence isn't just the absence of noise. It's not a vacuum. It isn't just nothing happening.
No.
Silence is an active, it's an expensive, and it's a critical decision that a cell has to make.
That is the hook for today. That's our whole mission. We are exploring the decision-making layer of biology.
Right.
And to do this, we are pulling from two absolute heavyweights in the field. We have Chapter 28 from Leninger Principles of Biochemistry, which is, effectively our Bible for the systems view, the deep molecular mechanics.
And the evolutionary conservation. It's the how it works book.
It's the how it works book, exactly. And we are pairing it with the section on gene expression from Harper's Illustrated Biochemistry, specifically Chapter 38, which brings the medical reality.
The pathology, the
pharmacology,
the cancer wards, the developmental disorders, the why this matters in a hospital perspective.
It's the perfect synthesis, really. Leninger builds the car, Harper's drives it to the hospital.
It really is. And looking through these chapters, I was struck by one central tension right away.
Yeah.
The default state of the human genome.
Mm-hmm.
It isn't go, it isn't maybe, it's stop.
That is the fundamental realization that changes how you understand your own biology. I mean, it changes everything.
How so? If every gene in your DNA were speaking at once, if all 20,000 instructions were being executed simultaneously, you wouldn't have a cell.
You'd have chaos.
You would have chaotic noise. You'd have immediate metabolic collapse and cell death. It would be a complete disaster.
So the mission today is to figure out this decision-making layer. We know for a fact that a neuron and a liver cell have the exact same library of DNA.
That's identical.
They have the same books on the shelves. But the neuron is reading the How to Conduct Electricity book, and the liver cell is reading the How to Detoxify Alcohol book. How do they know? Who makes that choice?
That is the question of gene regulation. We are moving from the chemistry of reactions to the chemistry of choices.
Okay, before we dive into the mechanics, and we're going to get into the weeds of zinc fingers and histone tails, believe me, let's just talk about the why.
Okay.
Aside from the chaos of everything happening at once, why is the cell so obsessed with keeping things turned off?
Economics. Pure and simple.
A cellular budget.
The cellular budget is incredibly tight. I mean, think about what we've learned in previous deep dives about protein synthesis.
Oh, it's expensive?
It's insanely expensive. It costs 2 ATPs just to charge a single tRNA. It costs GTP to move the ribosome one codon down the line. Making RNA and then translating that RNA into protein is the most expensive thing a cell does.
It has to be a huge chunk of the daily energy output.
A massive percentage. So if you're bacteria swimming in a puddle and there's no lactose around, why would you waste precious energy building the machinery to digest lactose?
You wouldn't. It's pointless.
You wouldn't. You regulate to survive.
Yeah.
If you spend energy on proteins you don't need, you get outcompeted by the neighbor who didn't. You die. It's that simple.
So regulation is the ultimate efficiency hack. It's like a molecular inventory management.
It is lean manufacturing at the molecular level. Don't build what you don't need right now.
Okay. Now, let's look at this default state concept again. You said for humans, the default is stop. Is that true for everything? What about our distant cousins, the prokaryotes? The bacteria?
This is where we see a massive evolutionary divide. And Leninger makes a very, very sharp distinction here between prokaryotes like E. coli and eukaryotes like us.
And this difference matters.
Understanding this difference explains a lot about why our diseases look the way they do. It's fundamental.
Okay, so set the scene for the bacteria. What's its genome like?
In a bacterium, the genome is relatively small. It's physically accessible. It's just floating in the cytoplasm, usually in a circle, and for the most part, the RNA polymerase.
That's the machine that copies the DNA into RNA.
Right, the transcription machine. It can pretty much just grab on and go. The DNA is naked.
Naked DNA.
Essentially. It's not wrapped up in complex protein structures like ours is. So the default state of a bacterial gene is generally on N.
So it's like an open plan office.
That's a great analogy. It's an open plan office where everyone is ready to shout their ideas at a moment's notice.
So if you want quiet.
If you want quiet, you have to actively go around and shush people. You need a repressor.
Yeah.
You need to put a physical block in the way of the polymerase.
This is what Leninger calls a negative regulation.
Exactly. The default is go. So if you don't want the gene expressed, you park a protein on the DNA that says road closed. You put a brick wall in front of the car, but if you take the wall away, the car drives.
Active unless repressed. Okay, so that's bacteria. What about us? What about eukaryotes?
We're the polar opposite. Our DNA isn't just floating around. We have a massive amount of it, about 1 meter or 2 in every single cell.
Which is just mind-boggling.
It is. And it is wrapped and coiled and packed into a structure called chromatin. We've discussed histones before.
Right, the little protein spools that the DNA winds around.
Exactly. In a human cell, the DNA is wrapped so tightly around these histones that the machinery, the RNA polymerase, it physically cannot get to the start line. It can't even see the promoter.
So the default state is OF.
The default state is OFF because the library is locked.
So if bacteria is an open plan office,
Eukaryotic cells are like the rare book section of a high-security library. The stacks are behind cages. You can't just walk in and grab a book.
you need permission.
You need a key. You need permission. You need a whole team of people to unlock the gate, find the book, and open it for you. This is positive regulation. You have to actively recruit the machinery to the site.
And the scale of this problem is just, Harper's threw some numbers at me that really put this in perspective.
It is staggering. A mammalian cell carries about 1,000 times more genetic information than E. coli.
1,000 times.
1,000 times. But here is the kicker, and this is from Leninger. Of the roughly 20,000 genes we have, only a tiny fraction are turned on in any given cell at any given time.
Because of specialization? A skin cell needs to be a skin cell.
Exactly. You have what we call housekeeping genes. These are things that every cell needs, like the instructions for how to build a ribosome, or the enzymes for glycolysis, or the proteins for the cytoskeleton.
The basics.
The basics. Those are the lights that stay on in the building all night. Every cell needs those just to exist.
But then you have the specialty genes, the luxury items.
Right. Hemoglobin. You need that in an erythrocyte, a red blood cell, at massive, massive quantities. But you decidedly do not want hemoglobin filling up your neurons or your skin cells.
It would be useless.
It would be useless clutter, or worse, it could be toxic. So regulation isn't just about efficiency like in the bacteria. In us, it's about identity.
Regulation is identity.
Precisely. A neuron is only a neuron because of the very specific set of genes it expresses and, just as importantly, the thousands of genes it actively suppresses.
Yeah.
If it loses that regulation, it loses its identity.
Which is a perfect segue into the how. Okay, we have this locked library. We need to get a book out. We need to turn a gene on. Let's talk about the personnel. Who has the keys?
We enter the world of transcription factors.
I love the name. It sounds so bureaucratic. The factors.
It does. But they are the movers and shakers of the nucleus.
Yeah.
Harper's defines them pretty simply. Proteins that bind to specific DNA sequences to either activate or repress transcription. But in our locked library analogy, these are the librarians who hold the keys. They know exactly which shelf, which book they need to go to.
But here's the math problem I ran into while reading Leninger. We have, 20,000 or so genes? Do we have 20,000 different transcription factors? Is there a specific turn-on gene number 405 protein?
That would be incredibly inefficient. Think about it. If you needed a unique regulator for every single gene, your genome would be cluttered with nothing but regulators regulating regulators.
An infinite loop of regulation.
It'd be an infinite loop. You'd run out of space. The cell is smarter than that.
So what's the solution?
The solution is a beautiful concept called combinatorial control.
Combinatorial control. Okay, unpack that for us.
Think about the English language. We have, hundreds of thousands of words? Do we have a unique symbol for every word?
No, of course not. We have an alphabet, 26 letters.
Exactly. You mix and match those 26 letters to create infinite meaning. The cell does the exact same thing with regulatory proteins. You don't need a specific protein for every gene. You use combinations.
So protein A plus protein B turns on the growth gene, but say, protein A plus protein C turns on the stress gene.
Correct, and it gets even more physical and complex than that. These proteins often work as dimers. They have to pair up to function.
Okay.
And Lineker shows the math on this. It's pretty amazing. A family of just 4 different proteins can mix and match to form 10 different combinations.
Oh, I see. Because you can have A with A with B, A with C, B with B. Wow, the math ramps up fast.
It ramps up exponentially. And the implication is incredibly subtle control. It allows the cell to say something complex instead of just a binary on-off switch. The gene can interpret a sentence.
A full sentence.
A full sentence. It can say, I will turn on, but T only if protein A is here, and D protein B is here, and D protein C is not here.
It's logic gates. It's biological computing happening right on the DNA.
It is. The promoter region of the gene has landing pads for all these different proteins. The gene doesn't fire until the right combination is assembled. It's like a nuclear launch code. You need 3 people to turn their keys at the exact same time.
Let's talk about that physical connection, because Lineker gets really into the 3D shapes. How does a protein actually read the DNA? It's not like it has eyes.
Right.
And the DNA is a double helix. The bases, the letters are on the inside of the staircase. How do you read a book when the book is closed?
That is one of the most beautiful mechanisms in biochemistry. The protein doesn't read it with eyes. It reads it by touch.
By touch.
It reads the surface of the DNA helix, like Braille.
That's a cool image. How does that work?
So if you look at a model of the double helix, it's not a smooth cylinder. It has grooves. There's a wide one called the major groove and a narrow one, the minor groove.
Okay.
These are like indentations spiraling up the helix. And the edges of the base pairs, the adenines, thymines, cytosines, and guanines, are exposed in those grooves.
Even though they are paired up in the middle, they're still accessible from the outside.
Yes, their edges stick out slightly into the groove. And those edges present a specific chemical landscape, a specific shape and charge distribution. An adenine-thymine pair looks chemically different from a guanine-cytosine pair from the outside.
You mean in terms of like hydrogen bond donors or acceptors?
A hydrogen bond donor here, a hydrophobic patch of a methyl group there. It's a unique pattern for each base pair.
And the transcription factors have shapes, these little protein domains that fit perfectly into those specific chemical patches.
Exactly. We call them structural motifs. You'll see terms in the textbooks like zinc finger, helix turn helix, or homeodomain.
The zinc finger always sounds like a villain from a 1960s spy movie. Goldfinger's cousin, zinc finger.
It sounds villainous, but it's actually a very delicate, elegant structure. It's amazing. It uses a single zinc ion to stabilize a small loop of protein.
So it's a literal finger of protein.
A literal finger that tucks right into the major groove of the DNA. The zinc isn't doing the binding, it's just the structural support.
It's the scaffold.
It's the scaffold. It holds the protein shape absolutely rigid so that the amino acids on the finger can make precise, specific contacts with the DNA bases. It feels the sequence without ever having to unzip the DNA.
It reads the code through the spine of the book.
A perfect way to put it.
Yeah.
And the specificity is breathtakingly high.
How high?
A zinc finger protein can distinguish its specific target sequence of, say, 8 base pairs from the other 3 billion base pairs in the genome. If it doesn't fit perfectly, if even one hydrogen bond is off, it won't dock.
That helps me visualize it. So these proteins scan the genome, they find their specific landing strip, and they dock. But wait, we established earlier that the library was locked. We said the stacks were closed, the DNA is wrapped in chromatin. How do they even get to the DNA landing strip if it's buried against a histone?
Excellent question.
Yeah.
You've identified the paradox. If the DNA is wrapped tightly around histones, the zinc finger can't get into the groove. It's blocked.
Right?
This brings us to section 3, access control.
The barrier.
The nucleosome. The nucleosome. To recap, DNA is wound around a core of 8 histone proteins. It's a very tight interaction. We were talking about fundamental electrostatics here.
Okay.
DNA is built on a phosphate backbone, which makes it incredibly negatively charged.
Right.
Histone proteins, on the other hand, are rich in the amino acids lysine and arginine, which have positively charged side chains.
Opposites attract.
They attract very, very strongly. It's like a powerful magnet. So if the promoter, the start here sign for the gene, is buried against the surface of a histone, the transcription machinery cannot see it. It is physically hidden.
So we need to either move the histones out of the way or pry them open somehow.
Or change their charge. And this is where Harper's introduces a concept that is just central to modern biology. It's called the histone code.
The histone code. Okay, that implies there is information written on the histones themselves, not just in the DNA sequence.
That is exactly what it is. The histone proteins aren't just inert spools. They have these long, unstructured tails that stick out from the core like the ends of a ribbon. And the cell can chemically modify these tails.
It can tag them.
It can tag them. It's like putting a do not disturb sign or a please clean room sign on a hotel door handle.
Okay, I like that analogy. So what's a common tag for open for business?
The most common modification for activation for turning a gene ON is acetylation.
Acetylation. So adding an acetyl group. Leninger explains the chemistry here, and it's surprisingly simple, but what does it actually do?
It neutralizes the charge. Remember, the histone holds the DNA because the histone is positive and the DNA is negative.
Right. The lysine.
The lysine residue on the tail has a positive charge. When an enzyme adds an acetyl group to that lysine, you remove the positive charge. You make it neutral.
So the magnet turns off.
The grip loosens. The electrostatic attraction is gone. The chromatin relaxes, it decondenses. The stacks of the library slide open.
Yeah.
And suddenly, the DNA is accessible. The transcription factors can see the landing strip.
Wow. And the enzymes that do this have these great names.
Harper's uses really intuitive terminology here that I just love. It makes it so much easier to remember. We have code writers.
The writers.
These are enzymes like histone acetyl transferases, we just call them HATs, that add the chemical flags. They write the open command onto the histone tail.
Okay, so you have writers.
Then you have code readers. These are other proteins that contain special domains. You'll see names like bromodomains that can spot the flag. They see the acetyl group and say, this region is open for business.
And they docks there.
They dock there, and they help recruit the polymerase and the rest of the machinery.
And I assume if you want to shut it down, you have erasers.
Exactly. You have code erasers. These are enzymes called histone deacetylases, or HDACs. They come in, they snip off the acetyl group. The positive charge returns to the lysine.
The magnet turns back on.
The magnet turns back on. The chromatin snaps back tight against the DNA, and the gene is silenced again.
This is wild because it means there is a layer of memory on top of the DNA. The DNA sequence itself hasn't changed at all. An A is still an A, G is still a G, but the accessibility of that DNA has changed.
You have just perfectly defined epigenetics.
Okay, let's unpack that word, because it gets thrown around a lot in pop science, right? Oh, you can change your epigenetics with kale smoothies and positive thinking. But what is the rigorous Harper's definition?
Harper's defines epigenetics very strictly, and it's a great definition. It is. Heritable changes in gene expression that do not involve changes in the DNA sequence.
Heritable is the key word there.
It's the most important word.
Because if I have a liver cell and it divides, the new daughter cell needs to know that it's also a liver cell. It can't just reset to 0 and become a neuron.
Exactly. If it reset, you just have a blob of generic cells after a few divisions.
Yeah.
The identity of the cell has to be passed on. The histone code is part of that, but histone modifications can be a bit transient. They can change. An even more stable mechanism, the permanent ink of the cell, is DNA methylation.
Okay, this is another layer. This is where we're not just tagging the histone proteins, we're putting a chemical cap on the DNA base itself.
Specifically on the base cytosine. The enzyme adds a methyl group, little CH3, onto the cytosine. We call it 5-methylcytosine. And it usually happens in regions where a cytosine is followed immediately by a guanine. We call these CPG islands.
And the general rule:
high methylation correlates with deep silence. If a promoter region of a gene is heavily methylated, it is generally locked down tight. It's the do not enter tape. It physically blocks transcription factors and also recruits those eraser proteins we talked about, the HDACs, to come and tighten the chromatin even further.
So how is this heritable? This is the part that always got me. When DNA replicates, the new strand is fresh, right? It's just raw nucleotides. It doesn't have the methyl marks.
That's the beauty of the mechanism. It's so elegant. So DNA replication is semi-conservative.
Yes.
You unzip the helix, and you have the old parent strand, which has the methyl marks.
Right.
And you have the newly synthesized daughter strand, which is clean. It's unmethylated. We call this state hemimethylated, half-marked.
Okay, so the old strand says silence, but the new strand is blank. That seems like a problem.
It would be, but there's an enzyme, a maintenance methylase, that trails right behind the replication fork. It's like a little proofreader. It scans the DNA. It looks for those half-marked spots.
And what does it do?
If it sees a methyl group on the old parent strand, it immediately places a new methyl group on the cytosine, on the new strand, right opposite it.
It's copying the notes. It's transferring the pattern.
It is copying the silence. It ensures that the liver cell suppression pattern is perfectly stamped onto the new genome. This is how a cell remembers who it is, generation after generation of cell division.
This has to have huge medical implications. Harper's talks about this in the context of cancer.
It does. It's a massive field. We often think of cancer as a disease of mutation. A gene gets broken, an oncogene gets permanently switched on.
Yeah, broken hardware.
Exactly. But Harper's points out that cancer is very often a disease of epigenetics. It's corrupted software.
How so?
Imagine you have a tumor suppressor gene. This is a gene whose job is to stop cancer. It's the emergency brake on cell division.
We want that gene on at all times.
We desperately need it on. But in some cancers, that gene isn't mutated. The DNA sequence is perfect. The code is fine. But the promoter has been hypermethylated.
The cell has accidentally put permanent do not enter tape all over the emergency brake.
Exactly. It's a quiet pathology. The gene is there, it's perfectly functional, but it's been silenced. This is why epigenetic drugs, like HDAC inhibitors or demethylating agents, are a huge area of cancer research. They're trying to strip those marks off to reawaken the cell's own suppression systems.
That is fascinating. It's not broken hardware, it's corrupted software. That's a perfect way to put it.
It really is.
So we have the default state, which is OOF. We have the keys, the transcription factors. We have the door, which is chromatin. And we have the memory, the epigenetics. Now let's talk about geography. The geography of the genome. Because this fact from Leninger just blew my mind. The switch that turns a gene on isn't always right next to the gene.
Not at all.
It can be thousands, sometimes tens of thousands of base pairs away.
Ah, yes. Action at a distance. We are talking about enhancers.
Enhancers. This feels so counterintuitive. If I want to turn on a light, the switch is usually on the wall right next to the door. I don't walk 3 houses down the street to flip a switch from my kitchen.
But in the genome, you might. An enhancer is a short sequence of DNA that binds to activator proteins. But as you said, it can be located way upstream or way downstream of the actual gene it controls.
So how does the signal get there? I mean, is it sending a wireless message down the chromosome?
No, it's even cooler than that. It loops. The DNA itself is flexible. The cell uses what we call architectural regulators, proteins with names like CTCF and cohesin, that literally grab onto 2 distant parts of the DNA and bend it.
They fold the chromosome.
They fold it into a loop, so that the enhancer, which is miles away in linear base pair terms, physically touches the promoter in 3-dimensional space.
So to go back to my light switch analogy, it's like the switch is on the wall across the room, but you use a really long wire to connect it to the bulb.
And the wire is the DNA loop itself. It brings the activator protein on the enhancer right next to the transcription machinery at the promoter.
And Harper's takes this a step further and talks about super enhancers, which sounds like a concept from a Marvel movie.
It really does. Super enhancers are massive clusters of these individual enhancer elements. They're all packed together, and they work synergistically to drive really high-level expression of the genes that determine cell identity.
The master identity genes, the ones that say, you are a T cell or you are a neuron.
Exactly. And what's fascinating is the physics involved here. This is cutting-edge stuff that Harper's is including. It mentions biomolecular condensates.
I saw that term. It sounds like physics, not biology.
It's where the two meet. It turns out that at these super enhancers, the concentration of transcription factors, polymerases, and coactivators gets so high they undergo a phase separation.
What does that mean? Like a drop of oil in water?
Exactly like a drop of oil in water. A liquid droplet of pure activation energy forms right around the gene. It creates a localized factory. It excludes the repressors and concentrates all the activators. It ensures that the gene is transcribed at incredibly high and sustained rates.
Wow. That changes how I picture the nucleus. It's not just a soup of molecules bumping around randomly.
It's not a soup. It's got these dynamic, self-organizing droplets forming and dissolving all the time. It's a lava lamp of regulation.
A lava lamp of regulation. I love that. Okay, so we've covered space action at a distance. Now let's talk about the 4th dimension. Time.
Temporal regulation. This is critical because a signal doesn't always mean the same thing. It depends on how long the signal lasts and how the cell is programmed to interpret it over time.
Harper's categorizes these responses into type A, type B, and type C. And I found this super, super helpful to categorize how our body reacts to things. Can you walk us through them?
Sure. Type A is the simplest. It's a direct correlation. The signal comes in, the gene turns on. The signal goes away, the gene turns off.
Like me eating a cookie. Sugar enters my bloodstream. The insulin gene turns on. Sugar gets cleared. The insulin gene turns off.
Perfect. That's homeostatic control. It keeps you balanced. Now, type B is a bit trickier. The signal comes in, the gene turns on. but then its expression fades, even if the signal is still there.
Sensitization adaptation.
Exactly. The cell adapts. It says, okay, I heard you, I've responded, I don't need to keep shouting. This happens with a lot of hormone signaling. You don't want to overreact to a chronic, persistent signal. It prevents system overload.
Okay. And type C?
Type C is the big one. It's the trigger.
Yeah.
The signal comes once, the gene turns on, and then it stays on forever, even long after the initial signal is gone.
That sounds like growing up. That sounds like development.
It is differentiation. It is how you become a tissue. Once a cell receives the signal to become a heart muscle cell, it doesn't need that signal telling it to be a heart cell every single day for the rest of your life. The decision is made, it gets locked in epigenetically.
And this leads us to the most mind-bending part of the reading for me, the developmental cascades in lineager, the fruit fly.
Drosophila, the classic model organism for a reason.
I always thought, who cares about a fly? But the logic is identical to how we're built, right?
It is deeply, deeply conserved. Evolution does not reinvent the wheel if it doesn't have to.
Yeah.
The way a fly builds its body from a single egg is a masterclass in hierarchical regulation. It starts with the mother.
The maternal genes.
The mother fly deposits mRNA molecules into the egg before it's even fertilized. These are the first instructions. They create gradients that define front and back, top and bottom.
So before the embryo even starts dividing, it already knows which end is the head.
Yes. A protein called B-coid, for example, forms a gradient, high concentration at one end, low at the other. That simple chemical gradient is read by the next set of genes, the segmentation genes.
And they do what the name says?
They slice the body into a series of repeating segments, like a little worm. Then come the master identity genes, the homeotic genes or Hox genes. These are the genes that decide what goes on each slice. This slice gets a leg, this slice gets a wing, this slice gets an antenna.
And the conservation here is spooky. You have to tell them the PAX6 story.
The PAX6 experiment.
Yeah.
This is one of the most famous and profound experiments in developmental biology.
Okay.
So PAX6 is a gene that is critical for eye development. Mice have it, flies have it, humans have it. It's a master regulator.
So researchers did a swap.
They did something even crazier. They took the PAX6 gene from a mouse, the instruction says build a mouse eye, and they inserted it into the leg of a fruit fly. And they used a genetic trick to force it to turn on in the leg cells.
And what happened? Did the fly grow a mouse eye on its leg, a tiny little hairy mouse eye?
No. That's what you'd think. It grew a fruit fly eye.
On its leg?
On its leg. A perfect, faceted, compound fruit fly eye.
That completely blows my mind. The mouse gene gave the order, build an eye. But the fly machinery, the downstream genes, interpreted that command using their own instruction manual.
Exactly. It proves that PAX6 is a master switch. It doesn't contain the blueprints for how to build an eye. It doesn't have the code for the lens or the retina proteins. It just knows how to shout the high-level command, build an eye here. The downstream workers, the fly's own genes, hear that command and build the only kind of eye they know how to build.
It shows that the logic of life is modular. You can swap out the generals, and the foot soldiers will still follow the orders according to their own training.
It emphasizes that these regulatory networks are hierarchies. You have generals, colonels, and foot soldiers. PAX6 is a 5-star general.
So far, we've talked about turning genes on and off, dialing the volume up and down. But sometimes, Harper's points out, the volume knob isn't enough. Sometimes the cell has to take extreme measures.
Yes. Standard regulation is like turning the volume on your stereo up to 10, but sometimes you need the volume at 100. Or you need to smash the stereo and build a new one that plays a different song entirely.
Okay, let's talk about gene amplification. This is when the cell actually makes more copies of the DNA itself.
Correct. For the most part, the genome is sacred. You get 2 copies of every gene, one from mom, one from dad, and that's it. But there are exceptions. Take amphibian eggs.
Okay.
They need to make millions and millions of ribosomes incredibly fast to support the rapid cell division of the growing embryo. The standard 2 copies of the ribosomal RNA genes simply cannot produce enough RNA fast enough.
So they photocopy the gene.
They amplify it. They create thousands of extra copies of that specific gene region, often as little satellite DNA loops, just to meet the overwhelming demand for ribosomes.
But there's a dark side to this, isn't there, in medicine?
Cancer. Once again, some cancer cells learn to use this mechanism against us. One of the most heartbreaking things in oncology is when a patient initially responds well to chemotherapy, but then the tumor comes back and it's completely resistant.
What happened?
Very often, it's gene amplification. Methotrexate is a common chemotherapy drug. It works by targeting an enzyme called DHFR. The cancer cells that survive the initial treatment are sometimes the ones that have randomly amplified the DHFR gene. They've made hundreds of extra copies of it.
So even if the drug knocks out 99% of the enzyme,
The cell has made so much extra enzyme that it doesn't care. It just powers through. It's an arms race, and the cancer uses our own regulatory tricks to win.
that is terrifyingly clever.
Biology is relentless. It finds a way.
Okay, another extreme measure is gene rearrangement. This is the immune system, right? B cells.
This is one of the few times our body intentionally and permanently breaks and rearranges its own DNA.
Because we need antibodies for everything, every possible bug.
We need to be able to make antibodies to recognize millions, maybe billions of potential pathogens, flu, COVID, bacteria, things that haven't even evolved yet. We
Okay.
can't have a separate gene for every single one. We don't have enough DNA in our bodies.
So we improvise, we cut and paste.
The B cells have a menu of gene segments, V segments, D segments, J segments. And as the B cell matures, it uses enzymes called RAGB proteins to physically cut the DNA. It randomly picks one V, one D, and one J segment and stitches them together. And it throws the rest of the DNA away.
It throws the DNA away permanently.
Gone. Deleted from the chromosome forever. That B cell has now created a unique gene sequence that codes for a unique antibody. It has a permanently altered genome.
So every single B cell in my body is genetically distinct from my liver cells or my neurons at that one spot at least.
At that specific locus, yes. It
Okay.
has committed to a specific identity by deleting all the other options.
That is the ultimate burn the ships strategy. There is absolutely no going back from that.
No going back. It's a terminal commitment.
Okay, before we move to the final clinical synthesis, we have to mention the RNA itself. Regulation doesn't end when the transcript is made.
No, absolutely not. That's just the beginning of the journey. The mRNA has to survive the dangerous journey to the cytoplasm and find a ribosome. Harper's talks about RNA stability.
The poly-A tail and the 5-foot cap.
Exactly. These are the armor. They're protective structures on the ends of the mRNA that protect it from being chewed up by enzymes called nucleases. Generally speaking, the longer the poly-A tail, the longer the life of the mRNA and the more protein you can make from it.
But the cell also has weapons to target that armor.
Oh.
I saw a mention of marinas? MicroRNA?
Yes, these are a huge deal in regulation. They are tiny, non-coding pieces of RNA, only about 22 nucleotides long. They are like homing missiles.
Homing missiles.
Each miRNA is programmed to match a specific mRNA sequence. It binds to it. And that binding event triggers a protein complex called RISC to come in and just slice the mRNA in half, destroy it.
So you could transcribe the gene, send the message out, and then intercept the message before it's read.
It's a way of fine-tuning gene expression. Or it's an emergency brake. If you realize you made too much of a protein, or if you need to shut down a pathway now, you send in the miRNAs. It allows for a much sharper, faster off switch than just stopping transcription and waiting for all the existing mRNA to degrade naturally.
It's amazing how many layers of control there are. It's not just one switch. It's a switch, then a dimmer, then a filter on the wire, then a shredder at the end of the line.
Redundancy and precision. That is the theme of eukaryotic gene regulation.
So bringing this all together, the so what of this whole deep dive. We've touched on cancer a few times, but let's look at the big picture of human health. Harper's makes a very strong case that many diseases are actually diseases of dysregulation.
Absolutely. The old way of thinking was to always look for a broken protein, a mutated enzyme that doesn't work. And that certainly happens. But increasingly, we find that the protein sequence is fine. The problem is dosage or timing.
You mean like Down syndrome?
Trisomy 21 is the perfect example. The genes on chromosome 21 are normal genes. They aren't mutated. They code for perfectly functional proteins. But there are 3 copies of the chromosome instead of 2.
So it's a volume issue. The dose is wrong.
It's a massive regulatory imbalance. The orchestra has too many trumpets. It throws off the entire symphony. The developmental delays and the physical traits we see are the result of that subtle but widespread gene expression imbalance, often 1.5 times the normal amount, for hundreds of genes.
And then on the flip side of all of these diseases, we have perhaps the most hopeful technology in modern biology, stem cell therapy.
iPSCs, induced pluripotent stem cells. This ties everything we've talked about today together in a beautiful way.
This is the time machine.
It really is. For the longest time, we thought differentiation was a one-way street, a ball rolling down a hill into a valley. Once you become a skin cell, you are a skin cell until you die. You are locked in by that DNA methylation and that chromatin structure.
But Shinya Yamanaka proved that wrong.
He did. In 2006, he and his team published a paper that just shocked the world. He identified 4 transcription factors. Just 4. OCT4, SOX2, NANOG, and LIN28.
The Yamanaka factors.
If you take a mature, differentiated cell, a skin cell from an 80-year-old, a cell that has been skinned for decades, and you force it to express these 4 factors.
What happens?
It unlocks the library, it strips off the methylation, it opens up the chromatin, it reverses the arrow of time.
The skin cell forgets that it's skin.
It forgets that it's skin, and it remembers that it can be anything. It reverts back to an embryonic-like stem cell.
Yeah.
And from there, in the lab, you can then coax it to become a neuron, a beating heart cell, a pancreatic beta cell.
That proves definitively that identity isn't a permanent structural change to the DNA. It's just a state of regulation.
It's an epigenetic state.
Yeah.
And because it's epigenetic, it is reversible. That is the entire promise of regenerative medicine. We can potentially regrow tissues using a patient's own cells just by hacking their regulatory code.
It's incredible. We are basically becoming hackers, figuring out the passwords to our own cellular operating system.
And we are just getting started. It's a very exciting time.
So let's wrap this up. We've covered a massive amount of ground, from bacteria to fruit flies to our own cells. What are the big billboard-sized takeaways from this deep dive?
Okay, if I had to boil it down, I'd say 4 things. First, the default state in humans is silence. Access is restricted by chromatin. You have to actively work to turn a gene on.
It's a system of no unless.
Exactly. Second, it's combinatorial. You don't have a unique key for every door. You have a complex combination lock of transcription factors.
Okay.
Third, there is a second code. The epigenetic code, the methylation and the histone modifications, sits on top of the DNA and provides memory and identity that can be passed down through cell division.
And
Fourth, regulation happens in 4D. It involves complex looping in 3D space with enhancers, and it unfolds through precise cascades in time, like in development.
And I'd add a fifth, the clinical reality. Disease is often just the right song played at the wrong volume or at the wrong time.
Or the wrong song being played entirely because a suppressor was silenced.
Right. I want to leave everyone with a thought that really hit me while I was reading Leninger. We tend to think of our genes as our destiny. You know, oh, I have the gene for blue eyes, or I have the gene for this or that. But looking at all this, the gene itself is just a passive string of letters.
It is. It's data storage. It's a hard drive.
The real magic, the real life, isn't in the data. It's in the retrieval. A neuron and a liver cell are identical in their stored data. The only difference, is the choice of what to express.
That's a profound way to look at it. We are not just what our genes are. We are what our genes do. And that doing is a constant, active, energy-consuming process of decision-making every second of our lives.
It's about responsibility. The cell has to take responsibility for its identity every single second. It has to actively work to keep the liver genes on and the neuron genes off.
And when it stops taking that responsibility, that's chaos. That's disease. That's death.
Heavy stuff, but absolutely fascinating. If you want to see what these loops and zinc fingers actually look like, and you really should because the molecular models are just beautiful, open up your copy of Leninger Chapter 28 and check out the diagrams of the Lac operon and the Hox clusters. They are amazing visual anchors that make all of this stick.
And Harper's has excellent diagrams on the signaling pathways and the chemical mechanisms of those histone modifiers we talked about.
Thanks for diving in with us. We'll see you on the next one.
Stay curious.
And stay regulated. Catch you later.